We're going to look at gated channels. These are ion channels that are gated, meaning opened and closed, in response to a particular stimulus. This is in contrast to leak channels that are generally not gated. In other words, they are always open and therefore leaking ions in one direction or the other, into or out of the cell, based upon electrochemical gradients of the particular ions. These channels tend to be fairly selective about the ions they allow to pass through, and their purpose is to change membrane potential in response to a particular stimulus. And those stimuli can come in a variety of forms. And so you basically use these channels to receive information or transmit information. Now at the top here, we have these channels in the unstimulated configuration, and at bottom, we have them in a stimulated configuration. Okay, so what are our categories here? On the left, we have our ligand or chemically gated channels. These are channels that are opened by the presence of a particular molecule that binds to that protein, causing the channel to open. Again, which way an ion will move through this channel will depend upon the electrochemical gradient of that ion. Next, we have voltage-gated channels. They're often found where we need to transmit information along a cell, like a neuron, or a skeletal muscle fiber that needs to be stimulated from end to end. And these voltage-gated channels are opened by, literally, a change in VM, or voltage of the cell. So changing the voltage of the membrane causes the opening of a channel. Here's our gate opening, right? This is our gate that's now not blocking our pathway. And so changing the membrane potential causes a channel to open, which causes a change in membrane potential, right? So voltage-gated channels are stimulated by the same thing that they cause, essentially, right? And again, they can't stimulate themselves, so they need input from another type, often associated or adjacent ligand-gated channels. Mechanically gated channels are opened by physical stress on the membrane, right? So think of tactile receptors, or the receptors that tell you about the fullness of your stomach or your bladder, right? Those are receptors that get activated once we get pressure or stretch of the cell membrane. Finally, we have temperature-gated channels. These allowed us to sense body temperature or the temperature of our environment to a degree, found in our skin and in our brain and other places, again, allowing us to temperature regulate our bodies. Now, there is another group that's not shown here because it's very specific. These are photoreceptive cells that are sensitive to electromagnetic energy, and so those are channels that are opened by the presence of light or absence of light in some cases. And then finally, there are channels called nociceptors, which are pain receptors. And they're actually not a specific type themselves, because they can be either mechanically gated or chemically gated, ligand gated, maybe even temperature gated at extremes, where we induce pain in response to those particular stimuli.